Hello, and welcome to the Sentinel Innovation Methods and Seminar Series. My name is Rishi Desai from the Brigham and Women's Hospital in Boston, and I will be moderating today's session. The US FDA Sentinel system helps address questions related to safety and effectiveness of marketed medical products. The Sentinel Innovation Center focuses on developing innovative methods to further advance Sentinel by leveraging new data sources, building new tools, and enhancing real-world evidence capabilities. Sentinel Operations Centers focuses on supporting the FDA in use of the Sentinel Common Data Model and associated modular programs to gather evidence to support regulatory decision-making. We launched this webinar series to engage experts and innovators in key areas related to the Innovation Center's master plan and Operations Center's methods work. With that, I'm pleased to announce today's speaker, Dr. Keith Marsolo. Dr. Marsolo is an associate professor in the Department of Population Health Sciences at Duke University School of Medicine and a member of the Duke Clinical Research Institute, DCRI. His current research focuses on infrastructure to support the use of EHRs and other real-world data sources in observational and comparative effectiveness research and public health surveillance, as well as standards and architecture of multi-center learning healthcare systems. He serves as an advisor to the Pop Health Data Shared Facility and as faculty lead for the Pragmatic Health Services Research Functional Group within the DCRI. Dr. Marsolo was trained with PhD in computer science from the Ohio State University with a dissertation on data mining, specifically modeling and classification of biomedical data. He's also an active member of the Sentinel Innovation Center community and has led multiple projects. And the project that he's focusing on today is also work that is conducted under his leadership through the Sentinel Innovation Center. So with that, I will hand it over to Dr. Marsolo with the title of his presentation being Identification and Mitigation of Source Data Mapping Issues in Structured Electronic Healthcare Data. Before we get started, I would like to mention that your lines are muted. If you'd like to pose a question, please type it in the Zoom question and answer feature. We will address the questions at the end of the session. Finally, this session is being recorded and will be made available through the Sentinel webpage, which can be found at sentinelinitiative.org. Without further ado, I will hand it over to Keith for this presentation. Keith.
Great. Thank you, Rishi. So today we are, I'm going to talk about our Innovation Center project, Identification and Mitigation of Structured Electronic Health Record Source Data Mapping Issues. And so this is somewhat of a work in progress, still ongoing, but we've got some very interesting results that we wanted to share with the community. So in terms of the motivation for the project, there's really two sort of main drivers for the work that we're going to describe. So the first one relates to the harmonization of electronic health record data. And so as many people may be aware, a number of kind of what you would consider to be standard data domains within the electronic health record, such as medication orders and laboratory results, are not captured in what we would call sort of standard interoperable formats. To use these data for research or public health surveillance, you first must harmonize them to a reference standard. And since you have to go through this harmonization process, it's important to have procedures that can help verify that this transformation is accurate. So if you're using these data for any kind of analysis, they mean what you think they mean. And then the second sort of challenge that we're trying to tackle with this work is really the completeness of EHR data. And so, we often talk about EHR data like it's kind of this single thing, when in reality, it can come from a number of different systems or sources. And so this could be a single enterprise EHR with multiple modules, or in reality, from many different systems that might have changed over time. A health system may have cardiology procedures that are sort of ordered through one system from 2017 to 2021, billed through a different system for part of that time, and then sort of a third system over the last couple of years. So you've got this this changing enterprise underneath, and so that affects sort of where the data may come from and what you might use for research. And so given that we have access to these sort of multiple streams of data, which could be sort of clinician entered information, orders entered into the system, billing data on the health system side, claims that were submitted to health plans, and so forth. Can we start to look at some of these different streams to help determine if we have, sort of a complete set of EHR data? And so the examples that I'm going to show in some of these initial slides are taken from the National Patient-Centered Clinical Research Network, or PCORNet. But really, the challenges that I'm talking about, they kind of exist regardless of the source. If you're working with EHR data, these problems apply. So there's nothing in here that's really PCORNet specific. Although, again, I am taking examples from PCORNet. So the first example that I'll talk about is the harmonization of medications in terminology called RX norm. And so within PCORNet, medication orders and administrations at most sites are coded to RX norm. And the reason for this is it's an interoperability standard maintained by the National Library of Medicine. And part of its purpose is to provide normalized names for medications, hence RX norm. But one of the features of RX norm, if you will, is that it can represent medications at various levels of granularity. And so if you look at this example on the slide, what you can see are essentially the second row there are what are known as RX norm term types. And so these are different ways of representing a medication. And you can see sort of from the middle columns there that sort of the different term types encode different aspects of a medication. And so if you've got a medication in a string shown in the sort of top right corner highlighted there in yellow, sort of the original string, and then you can see how that might be represented in RX norm at the various term types. And so, I think what's important here is, again, there's some information loss as you go down the rows. And then the other thing is that some of the term types require multiple records to represent a given sort of medication. If it's a multi-ingredient drug, you're going to have sort of multiple records in your system. And the reason why, we sort of so this is with PCORNet, and one of the questions might be, so if I'm wanting to use sort of EHR data in an analysis, and I decide, I want to use a different terminology. Maybe I don't like RX norm. Why should I necessarily care? Even if a network like Sentinel wanted to use a different standard to represent EHR-based medications, it's quite possible that a data partner may need to transform, from RX norm to whatever the target is. And so, we felt that this is an example why it's important to understand what's going on. Now, within PCORNet, we've set up different tiers in terms of sort of what are most preferred, and you can sort of see them in the right-hand column. But again, what we see is that depending on sort of how data partners have transformed this information, they may not always be able to map to sort of the most preferred tiers. There may be reasons why some subset of their medication data are mapped to sort of a less preferred term type. And so then just trying to understand a little bit more around why that might occur. And to just give an example of sort of how this could have ramifications in an analysis, what we've done is, this is an example from a few years ago where looking at, this is all data from within a single data partner. And we looked at their medication records, and sort of what we looked at were the highest volume records by sort of RX norm code. And so those are the 4 columns on the left. And then we looked at the highest volume medication records by sort of the name, sort of the raw string in the system, and just compared them sort of kind of line by line. And what you can see in, and then we sort of, based on the numbers, we also calculated the percent agreement. And what you can see in sort of the lines with the yellow highlighting, if you take the medication names sort of on those right set of columns, you can see that they're in many cases representing multi-ingredient medications. But then if you look at the corresponding RX norm term on the left, it's actually been mapped to a single ingredient RX norm code. And so then this could indicate sort of, you know, a loss of information or sort of a mismapped medication. And so if you're trying to use these data in your analysis, you know, there could be sort of ramifications in it. So this is an example when we talk about harmonization issues. This is one of the things that we you know, we worry about in just trying to understand sort of the quality of the information. To give an example on sort of the completeness completeness issue, sort of looking at differences in rates of data provenance. And so this is looking at the rates of sort of the number of orders versus the number of medication administrations. And so this is actually looking at multiple centers. So each sort of dot or little cross on the graph represents the ratio of orders for a data mart. And really the purpose of this example was to just sort of look at the rates over time. And then what we can see is that in a few examples sort of highlighted there with the red boxes is that the rates are much higher than or much different than some of the other centers. And so this is an example where you know, there could be sort of an issue at play, such as, you know, sort of drop if the ratio of medication administrations versus sort of orders is much higher, you know, maybe some of the orders are missing or something like that that could be causing a problem, and sort of the other way around, you know, there could be medication administrations that are missing. So again, I'm just trying to look at some of these differences in different sources of data to indicate that there might be a problem. So sort of that's kind of the motivation, and that led us to our project goals and sort of objectives. And so the objective one was really around this data harmonization. So assessing the mapping and harmonization of structured elements to reference terminologies. In this case, we're specifically looking at lab results and then medications, both orders and administrations in an outpatient and inpatient settings. And then sort of work that's still in progress is really characterizing the severity of issues that are uncovered. And then objective two are metrics, although in you know, sort of in terms of implementation, they're just queries that are going to allow us for comparisons across complementary data domains. So specifically, we're looking at profiles of records across time, care setting, and provenance for a different condition, for a given condition to help us identify issues with data completeness. So sort of jumping into you know, some of the logistics and the results. So around objective one, the approach was to develop queries. So again, assessing mappings of medications in lab. In this case, we just limited ourselves to the top 200 by volume. In reality, across a given health system, you know, they may have several thousand unique laboratory tests and you know, thousands and thousands of different medications in their system. And so for each medication in sort of lab, we generated statistics on all the different combinations of structured fields and raw fields. And then this is sort of on the screen here is just an example of, you know, what for a given raw medication name, the different RX norm codes that could be it could be mapped to. And then this is an example where, you know, we're looking at sort of the dose, the unit in the common data model. In this case, it's mapped to other. And then looking at sort of the raw dose to try to understand, you know, sort of is there any commonalities? You can see in this example, milligram of salt, which is not in the value set of the common data model. You know, it's sort of highly used across the system. And so this is just sort of an example of what you can find out with the data. The queries that we ran were written for the CoreNet common data model, but certainly they're applicable to other common data models. And then the evaluation that is sort of still ongoing is, you know, looking at medication codes and tests associated with more than, you know, sort of one name. So you've got a single code, and then in the system, does that map to sort of multiple raw names in your common data model, and sort of the inverse of that. Looking at the concordance between the lab name and the medication, and then sort of ingredient. So, you know, does and the code. concordance between discrete fields, and then using the results to characterize issues by severity. And so just jumping into some of the initial results, we ran these across 3 different data partners within PCORnet. So the first thing that we looked at here was sort of looking at the raw medication system, and then sort of the RX norm codes that it's mapped to. And so the first thing was just the number of RX norm codes associated with a given raw name. And we did this for prescribing and then in the administration table, so orders and administration. And our assumption was that it really should be 1 for the most part. And we can see that the median result is actually is 1, so that's nice to see. But there are sort of cases where it's mapped to more than 1. And so these are a couple of examples here on the right side, where you've got, again, a raw name at 2 partners sort of mapped to more than 1 RX norm code. And so the first one, sort of the top 2 rows there on data partner 2, what you actually see is sort of a medication of naloxone, and the first row there, it's mapped to a blank RX norm code. So there sort of is no RX norm code assigned to that record. So that means sort of none of those medications are going to show up in an analysis. They're essentially missing from the analysis. And then the ones that we see on the sort of the bottom 2 rows, it's basically an example where the raw medication name sort of only encodes to the level of the ingredient. And then what you've got sort of with the RX norm code that's assigned is essentially a code that encodes more information. And so you essentially have a more granular code given the source data that you have. And this is actually one of the things that we see with some EHR data in some cases, where sort of the raw information is sort of at the ingredient level. And so there is a question of, when you have codes that encode more information, is that a potential issue for an analysis? Sort of the second thing that we wanted to talk about here is just essentially looking at it the other way around. So if we start with an RX norm code, and then we look at the number of sort of raw names that are associated with it. Again, assumption is that it should be 1, but what we can see here is sort of the median is 1, but the minimum and the maximum, there's sort of a very a bigger spread. And again, just some examples sort of what you see here. So what we've got, the way to look at the sort of the tables here is for each of the data partners, we've got the RX norm code, and then sort of what is the name or the description that's sort of associated with that RX norm code, and then the sort of 4th column is the raw name in the prescribing table, and then sort of the medication administration data is there on the bottom table. And again, some of these issues are likely okay. Sort of for data partner 1, you've got essentially RX norm codes that are less granular than the sort of the raw string in the in the source system. For data partner 2, it's an example where you've got a medication that doesn't have an associated RX norm code. For the sort of data partner 3, you've got some codes that are sort of overly specific, and then sort of the 5th one down, where you've really got, in some sense, it's a sort of multi-ingredient drug, although the other ingredient is dextrose, where you've only got sort of a single ingredient in your RX norm code. And then for the medication administration data, this is where we see you've got sort of RX norm codes for sort of sodium chloride in many cases. And these are examples where, basically if you've got a multi-ingredient inpatient mixture drug, and you're creating sort of multiple records for each of those, that's sort of that would be fine. But if that's not the case, you've got sort of mapping errors here. And so that's sort of examples that we see. And then the bottom 2 rows of the sort of the medication administration table, this is where there's sort of no raw name in the source system for this particular data partner for their administration data. So we actually have no way of assessing whether that's correct or not. So those are some of the things that we sort of see in the data. And then the other thing that we looked at, or that I'm going to sort of share today, related to sort of this first objective, was really looking at the concordance between ingredients. And so what this looked at was we sort of created clean versions of the ingredient that was associated with each medication, and then the ingredient that was sort of specified within the RX norm code, and just tried to look at the comparisons there. And you can see, sort of the spread of the results. And so as I'll show in sort of the next slide, for data partner 1, it's again this issue of a single inpatient medication mixture that's potentially represented by 2 or more records. Data partner 2, again, sort of multi-ingredient medication listed as single ingredient, and then with data partner 3, again, for administration data, there's no raw names, just numbers, and we sort of have no way of interpreting that. And so this is just an example of some of the things that we see, where you've got sort of the raw names are on the very left side. And so, the first 3 sort of rows there, where you've got some IV infusions that are being that are records there, sort of the RX norm ingredient that they're associated with is water, which is probably fine. But it's tough to say for sure. The next set of sort of records in those table are where they've got, again, there's ingredients that are actual medications that are associated with the ingredient of sodium chloride. And so if there's sort of secondary sort of multiple records for each of these medications, then that would be fine. But if there's not, that's an example of a mapping error. For the sort of last, for the sodium phosphate there in the middle, for data partner 1, it's an infusion. It's sort of mapped, sort of the associated code is dextrose or glucose, and so that can be delivered as part of the therapy that's specified there, but it could also be a mapping error. And then for the IMS template, mapped to a specific medication, that one appears to be an error. And then sort of the next set for data partner 1, it's sort of the nutrition, medications related to total parenteral nutrition, and then sort of they're mapped to ingredients that are associated with that. But again, if there's sort of not those multiple records, that's the problem. And then data partner 2 has sort of a raw prenatal vitamin just mapped to iron. And again, so if we don't see these sort of multiple records, then that could indicate a problem. So those are the types of things that we were sort of uncovering with our medication analysis here. Moving on to our second objective, sort of metrics to generate comparisons along provenance. And so sort of part of the motivation for this work is sort of the example that we show here on the right side of the screen. And so what this is an example of the kind of thing that we see in networks like PCORnet. So we're looking at sort of the percentage of patients in a cohort with a result for a given lab, a given lab test. And each bar represents the percentage of patients in that cohort sort of by a data mart. Now, on the sort of the very far right, you see there's a handful of sites within the network that have zero results, and so that's relatively easy to identify. That it's likely that they just haven't loaded any of those data into the data model. But if you look at sort of the percentages in the middle that sort of range between 5 and 60 or 5 and 80%, the question is, does that represent sort of missing results or practice variation? And so this is the kind of thing that we're trying to understand. So you can think of the same thing around percentage of patients in a cohort with a diagnosis for a certain comorbidity or something like that. So what we tried to do to address this was say, let's develop some queries that allow the comparison of records, on time, care setting, and provenance to identify potential issues. So the way that we operationalized this was to work with FDA for 4 specific conditions of interest that they wanted us to tackle. And then within each of those conditions, again, with FDA, defining associated diagnoses, procedures, medications, and lab tests. And so we went up to 8 in each of those categories. And then distributed a query package to partners to generate summary statistics stratified by year. So we looked at 2016 to 2021. We looked at encounter type, and then provenance. And so for the results that I'm going to talk about here, it's essentially the clinician entered information and then EHR billing. And really the focus of our analysis was within data mart comparisons, but cross data mart comparisons can still be informative. So we sort of able to do both with this type of query. And I'm not going to dwell on this. I just wanted to put it up here to just give some examples of the types of things that we looked at. Now, I said that we would look at sort of 8 things within each category. And I think if you're quick with the counting, you can see in some cases we had more, and in some cases that was where we had sort of code lists already predefined that we could essentially just add, quote unquote, for free for the query. So that's why some of these, particularly with labs, you can see we have sort of more than 8 in a category. There's no way that I can sort of go over everything that sort of came back within these queries, but the main thing was to just give an illustration of sort of what's in there, and then just sort of highlight the conditions that we looked at with these with these queries. So we looked at chronic hepatitis C or Hep C, chronic kidney disease or CKD, and then we looked at chronic obstructive pulmonary disease and pulmonary arterial hypertension. So those were the examples that we looked at as part of these queries. So in terms of the data partners, and I could have put this for the objective 1 results, in terms of background information, sort of large academic medical centers, all using Epic as their enterprise EHR, but they've all sort of switched from other systems in the last 10 years. And I think what's important is when we get into sort of some of the provenance sort of description. So data partners 1 and 2 populate sort of clinician entered or sort of ordered diagnoses as well as billed diagnoses. For procedures, data partner 2 has ordered in billed procedures. Data partner 1 only has billed procedures, and then data partner 3 sort of only sort of billed diagnoses and billed procedures. And what's important, I think, there's sort of a variety, sort of looking at sort of what's available. And when we look across ProcoreNet, which is a network of around 60 centers, you can actually see that there's quite a bit of heterogeneity in terms of sort of the partners and sort of the provenance of the information that they have available. And so again, part of this exercise of this analysis is to understand, again, when we start to look at sort of data provenance, what kind of information does that tell us? And then do we maybe need to revisit sort of as a community, whether or not we need to really pay attention more of these sort of these different aspects of data provenance. So, in terms of the results that we have, so we summarize this, for each condition by comorbidity, procedure, medication, and lab, stratified by encounter type and provenance by year. When we're looking at sort of the different encounter types, I think one of the things to note is that telehealth is grouped into the other category, and then for ProcoreNet purposes, or for the purposes of this analysis, it's lumped in with observation stays and institutional consults. And the reason I mention that is because in some of these graphs, you'll see the rate of other go up over time, and that's really because of the explosion in telehealth due to the pandemic. And then within the Epic EHR, sort of emergency department visits that lead to inpatient admission, they're treated as a single encounter. So in a lot of the common data models, you'll have sort of ED visits separated from sort of inpatient visits. They're treated as different encounter types. Within Epic, that can be hard to split. And so within PCORnet, we created a single encounter type that sort of encapsulates those ED to inpatient encounters. And then there's also within the results I'm going to show, we have what we call an other ambulatory visit, which is sort of lab only visits, refill encounters, and then there's a variety of sort of other ambulatory types that are there. And then with provenance values, the main ones to focus on are sort of the first two there, the OD, which is sort of order clinician entered, and then BI for EHR billing. We also within PCORnet, the values we've defined are claim and sort of derived, which would be sort of extracted via natural language processing. None of those are used in any of the data partners that responded to these particular queries, but they sort of do exist within the data. And then to improve, as a typo on my part, to improve readability, we've removed some of the rows that are sort of not a focus of discussion in some of these tables. And so now I'll go through a few different examples of the types of things that we're seeing, and then that we're going to work to try to investigate a little bit more as to what some of the issues might be. So this is an example. So what this is a data partner 2. This is looking at sort of within the CKD cohort, looking at a comorbidity of dyslipidemia across time. And what you can see is a huge drop in the percentage of ED visits between sort of 2016 and 2017, and then it sort of stays low. So 10% of the visits in 2016, and then essentially 0 or 1% of the visits in 2017. And so that could be an example of sort of just sort of a loss of ED visits. In this particular case, it might actually be also due to sort of an uptake of the that sort of inpatient to emergency visit, or sorry, emergency department to inpatient encounter type. So that might be a reason for the change, but sort of an example of the type of thing that you can sort of see with these data. Another finding, sort of a really interesting finding that we're still trying to understand is sort of provenance rates that differ by comorbidity. So this is a data partner 1 with, again, looking at the CKD cohort. And then what you can see are sort of the rates for diagnosis. These are sort of comorbidities, in this case, anemia, pericarditis, uremic bleeding, and then metabolic acidosis. And then we've got it for sort of the ordered and the, or the clinician entered, and then the billing. And what you can see is that the billing rates are, sort of relatively high, as you might expect, and relatively stable. But then what you see are very, very different rates with ordinance. And so, the question is sort of, are these comorbidities essentially added through the coding process, the medical coding process, and then sort of comes out of the bill? So, that might be one aspect of it. But I think, the other thing that we were sort of struck by is, we do have, like within PCORnet, and again, sort of many other sort of networks or projects that are leveraging EHR data, about a third of the network only had sort of order information for diagnoses. And so if you're only looking at sort of the clinician entered diagnoses, you may be missing a huge subset of your comorbidities, if that's all that you look at, sort of if these trends hold, and they do sort of appear to be the same across sort of data partner 2 as well. So that was sort of one thing that we were struck by. And then so that a takeaway from this this finding would be so like, well, we would have definitely rely sort of the billing sort of EHR billing data are kind of the way to go. And what we see is that's not always true. So this is an example sort of with COPD at data partners 1 and 2. And so this was looking at sort of comorbidities of asthma, and then sort of the billing provenance you can see. And then what you see is that sort of over time, the rates of sort of the billing provenance to sort of capture the entire cohort, it drops. It's not, it's not horrible, but you're sort of missing roughly 10% of your population. And so this is, again, sort of the flip side of it, where if you're relying sort of only on billing data, or sort of EHR billing data, it's possible that you're sort of missing some patients from your cohort. So it's just sort of one of the things to understand. And then when we look over at sort of data provenance for procedure data, so this is looking at data partner 2. This is where we see actually some pretty variable rates of provenance for procedure data. And so this is looking at sort of the table on the left is sort of CKD procedure information, so kidney biopsy, dialysis, and paleography, and then on the sort of right side of the table for PAH, echocardiograph, and then VQ scans. And what you can see is pretty large variations, ranging from essentially 100% from the billing information, down to 75% in some cases, and have some other examples that I couldn't necessarily show due to low cell sizes where the rates are even lower. And so again, if you're sort of just relying on sort of the billing information, you could have some missed records in your analysis. And so again, this is one of the things that we were struck by when we were looking at these results. And so now I'll sort of transition over to medication data. And so here, this is where we saw some very striking findings. And so when we're looking at sort of the ratio of administrations to orders, it's not that we expect, we know that, the sort of what's ordered in the system includes sort of medications that are dispensed at home, sort of orders that are sort of pended for an inpatient stay that are never filled. There's sort of information, the orders in general, we expect it to sort of be the superset of kind of everything that's entered into the system. And then, administrations is going to be a subset of that. At some places it's going to be sort of just inpatient administrations. Others may have some outpatient administrations that are sort of, you come in for, you come in and get a shot at in the sort of clinic at a well visit or something. It could have some of theirs. So it's not that we expect 100% or anything like that. It's really more like, do we see the rates being stable over time? And then, sort of big changes potentially indicate a potential problem. So in some cases, it might be influenced by changes in practice patterns. So, you used to give a first dose of a medication in the clinic so you could see how it's sort of tolerated, and then that's no longer standard practice. So those records sort of disappear. But, can we look at some of these changes, and then that gives us something else to investigate. And so just a couple of examples, sort of, here sort of highlighted on this slide. So this is sort of percent ratios of CKD meds at data partner 2. And so for the class of meds, the ESAs, it's close to 100%, and in some cases more than 100%. And so, again, that's not always a problem that would occur, but that would be something that we would potentially want people to investigate. And then this example is sort of the iron medication, where you've got, sort of starting close to 100% and then dropping to 27% over time. So just, sort of wanting to understand, are there sort of missed records there? And it's a very sort of busy slide here, but I think this is an example where, I think we're for sure, potentially seeing some dropped records. And so what you see are, this is for COPD meds, and then for, penicillin, if you're looking at the ratio of administration to prescribing, is sort of 9,000% in some cases, which, it looks, it just looks very, very strange. And then cephalosporins, another, 1,000%, sort of things like that. So, many, many, many more administrations than what you see in the orders table. And what's interesting with this is if you start to look at sort of the percentages of sort of medications at sort of data partner 1 and data partner 2 for sort of COPD. When you look at sort of the order information, which is sort of the table in the middle, you can see that again the percentages on the left for data partner 1 are much higher than what you see at data partner 2. So like again 0% for the penicillin and 1%, 2% for the cephalosporins. If you look at the bottom table for the medication administration data, you actually see that the percentages start to look a little bit more in line with one another. And so this is sort of an indication that there might be something with the order data for data partner 2. It's either that those records are missing or that they're sort of coded to something strange. So like it could have been an example where they're not mapped to a RX norm code or something like that. And we did use the same codes across both tables. So it's possible that their order data is coded to something else. But again, it's an indication that there might be something going on and something else to investigate with the records that you're seeing. So just a couple more slides here, and then we'll sort of have some time for questions. So just things for labs. So example with labs. So with labs, sort of the provenance information is not, it tends to all be sort of, ordered. There's not sort of a billing kind of feed for the most part from lab data. But this is an example where if you're sort of looking at the encounter types over time, you can see where essentially they went from 100% missing in 2016 at data partner 3 to, sort of much more well populated, so that there's only 4 or 5% missing in 2021. And this is really more probably less around data quality issue, if you will, more about sort of informing your sort of analytical plan. So looking at records by date instead of encounter type, and sort of not assuming that that the labs are in any way sort of necessarily tied to an encounter type. And then another sort of use of this type of analysis is really sort of looking at some of the rates of test completion. And so you could certainly look, for within your system. Again, the example that I showed sort of earlier, when you sort of have complete missing records, could just be an indication that they haven't been mapped to the common data model. So that's sort of the example on the left. And then the other thing that you can look at, sort of you see with sort of data partner 3 is starting to look at, again, do you see sort of tests that should be ordered together that are sort of typically part of the same panel? Do you start to see very similar rates for those tests within your cohort? For the example, for the data partners that we had for this particular study, I think actually we saw that was to be the case, where things that should have been ordered together were actually ordered together. But that's an example of the type of analysis that you could generate from this type of query. So just sort of summarizing here, this was sort of a very initial sort of step into this kind of topic. I think we were, we expected to find issues. We weren't entirely sure what we would uncover. But we did, even just looking at sort of a handful of data partners, I think we surfaced several findings. We're still summarizing sort of the issues that we found, and haven't really had a chance to try to run these to the ground to figure out what the issues might be related to. But that's sort of some of the next steps that we have here. But I think some of the takeaways, so that we know that there's sort of mapping issues that exist in the data, and sort of, looking at kind of the raw information to assess harmonization can help identify issues. But I think one of the things that's important is really setting expectations that, sort of the data partners properly populate these raw fields, and then, that you're actually going to use them as part of your quality assessments. And so, we do a lot of work within Coordinates. I work in the coordinating center there. And, it's was one of those things where, you know, the raw fields are created. I know other data models have this as well. They're there. They're kind of a nice to have, but it's not something that people enforce. And this might be something where, you know, it's have to be a little bit more strict in terms of what people populate there. And then similarly, one of the things that we're seeing is sort of having access to multiple streams of sort of data provenance can help raise issues, but, you know, really only if they're present. And so again, you know, so having worked on many of these systems, it's not necessarily the case that there's an expectation that everybody sort of populate all possible, you know, data streams. And I wonder, you know, do we need to move as a field to really populate them, because they are proving to be important. And so, you know, I think, you know, in the meantime, you know, we can sort of information about potential gaps can help inform study teams as they assess data for data marks or sort of single streams of provenance. And then, you know, beyond just sort of what we're seeing in some of these quality issues, is just like with the lab example, you know, helping inform analysis plans by surfacing, you know, rates of change and sort of encounter types and other things over time. Just want to acknowledge the study team and all the help that they've provided, and our coordinate data partners that participated in this. And I think with that, I'm happy to take any questions.
Great, thank you, Keith. That was a really excellent overview of your work over the years in trying to shed some light on the underbelly of the EHR data, which can be quite messy and complex to deal with. So thank you for your work, and thank you for this presentation. So I'd like to remind folks that this floor is open for questions. Please type them in your Zoom question answer feature, and we will address them as they come in. And as people are thinking about questions, I can probably start with one. So Keith, in your introduction slides, you mentioned that you were focusing on both RX norm and LOINC as well to see mapping issues, or to document mapping issues. And look like for RX norm, for the most part, the mapping was more or less accurate across the data partner sites, except some unique examples that you highlighted there. But I was wondering if this was more inconsistent with LOINC, or did you have similar findings there as well?
Yeah, so we're still sort of running some of those analyses. I think some of it, I'll just say sort of anecdotally, some of the LOINC issues that appear LOINC, it tends to be, so part of this, I think, is the fact that we sort of limited ourselves to the top 200. In some cases, like I know I showed at the very, very beginning, sort of, you know, some potential issues, but there's sort of more eyes on the stuff that's higher volume. And so, you know, those may be more likely to be sort of more well mapped. And then, you know, sort of on the long tail, it's a problem. But I will say that the medication mapping, sort of the process is different, in the sense that in many cases within the EHR, there's a medication database, you know, that a health system may pay for from, you know, I don't know if I want to name the names, but there's sort of a database that they may pay for, and there's within there, they may have sort of mappings to multiple terminologies that might exist. And then it's a matter of the analyst, when they've got their EHR, you know, sort of medication, they have to look into that database to sort of find the appropriate row, essentially, and assign that code. And again, certain medications are sort of different. With like LOINC, you know, sometimes that may be done through sort of like an HIE may actually provide a mapping back to a health system. We've seen it where sometimes it's part of the instrument that you know, somebody runs on the test. It'll have the LOINC, or you know, the you know, LOINC does provide some tools, but that tends to be, there's sort of more of a manual review in sort of how that's happened as opposed to the case of sort of medication. So so I think so part of this, I think what ends up happening is the medication example may be more sort of automated and it's more systemic, and then once you fix it, it sort of propagates, versus sort of LOINC, where in many cases there's a mapping table that somebody creates at some point in time, and then it can be more of a kind of a static thing. And so I think some of that is sort of influencing what we see in the data.
Okay, thank you for your insights there. There is one question in the chat box that just came in. This is from an anonymous attendee who asks, would analysis using the data before it is translated to the CDM, for example, using RxNorm or LOINC, something like that, encounter the same quality issues that are found after the translation?
Yeah, it's a good question. So I mean, essentially, yes. It would sort of have the same problem, right? So if like the lab data, for instance, at many health systems, you'll just have like the short name, or you'll have WBC as your white blood count tests, and so you basically have to go through this manual exercise, as I was saying, sort of say like the white blood cell tests that we have are this LOINC code, and there may be sort of 40 different LOINC codes for the white blood cell test, and over time the method may have shifted, so there's potentially multiple codes and sort of things like that. So that's like whether you're sort of picking up a data model that's somebody's already harmonized, or you're sort of doing it from scratch, like that exercise is the same. And then similarly for medication data, again, because it's not generated from the EHR in a reference terminology. It's sort of stored as a internal medication ID that's sort of unique to the EHR. You still have to go to that exercise of sort of mapping it to either NDC, if you know how it's dispensed, sort of RxNorm, or GPI, or sort of any of these things. There's a mapping exercise that needs to occur. And so whether you're doing it yourself or sort of assessing what somebody else has already been done, sort of the process tends to be the same.
Thank you. So there are no more questions in the chat box. I can probably follow up with one more question. So your findings around this provenance exercise were also quite interesting, where it seemed like for many examples that you were showing, the billing records appear to capture more comprehensively, or at least more frequently, the diagnosis that you were interested in. So I was wondering if you had done any kind of qualitative investigation into why, in some cases massively, the provider entered information is under recording this type of information, which seems counterintuitive, because so you would expect providers to kind of capture some of these history type variables with some completeness. So I was wondering if you were surprised, and if you did any kind of qualitative investigations around that.
Yeah. So that first, I was definitely surprised, and we haven't yet done that sort of qualitative exercise, and it's really out. Unfortunately, it's kind of outside the scope of this project, because it was really just, we're going to run the queries and sort of report back on what we find. I think the nice thing is now we have sort of something else to check next, and sort of some future work with this. I don't know. We will sort of go back to the data partners and try to understand potentially a little bit. I don't know if some of this is a function of like within the EHR and the providers entering, diagnosis information. I think in some cases they may be limited to a certain number, and then there's sort of what happens on the medical coding side, where they're looking through the unstructured text potentially and other things to sort of pull out some of these diagnoses. So that might be part of it, but that was something I think that we were really struck by. And again, just knowing sort of what we see like in networks like PCORnet, which, again, it's not, this is not limited to PCORnet. If you sort of do work in Odyssey or some of these other networks, they tend to be the same sort of academic medical centers and data partners, where a third of the network only has that order information. And so if you're using the data from those places, you might be massively missing things. And so, just trying to understand how to identify that and sort of what kind of mitigation strategy, I think, is sort of one of the pieces of future work for this this type of exercise.
Yeah, that is a great insight. And I think these types of quality metrics are going to be more and more important, and as we try to bring in diverse EHRs, and in some cases through aggregators, right, which may be aggregating a variety of EHRs into their system using their own rules and things like that. So yeah, that's great. So I think that there is no additional questions in the Zoom box. I don't have any more questions of my own. But really want to thank you, Keith, so much for sharing your findings with us. I think they are extremely relevant and important for us to consider as we try to bring in more EHRs in the Sentinel network. So thank you. And just as a reminder for everybody on the line, this session has been recorded and will be made available through our webpage. You can also find details and registrations for our future talks on the same webpage. So this concludes today's session. We hope to see you again next time, where we have another speaker describing some quite relevant issues related to EHRs and drug outcome investigation. So thank you everybody for joining in, and that's all from us.